Hello, and welcome to the first segment of 5 lecture recording segments on the nervous system. So in segment 1, we will get through part 1, slides 1 through 82. And in the first segment today, we will get through the first 6 topics shown here. So we'll start at the top, and I'll take you through the structure and function of neurons, and that will bring us to slide number 82. And then in the second segment, we'll pick up at the 7th topic shown here about postsynaptic potentials. We're going to focus our attention on the nervous system. It is important to acknowledge, to recognize, to remind ourselves that there are 2 organ systems that are dedicated to cell-to-cell communication in the body. There's the nervous system and the endocrine system. And the concept of cell-to-cell communication was introduced very early, lecture number 1 this semester, when we talked about the components of a feedback mechanism. At that time, I mentioned that there are 2 methods of communication between those components, either nerve impulses, sometimes called electrical signals, or chemical signals. And so we are building on that, and we can refine our understanding of what those things mean. So the nervous system conveys high-speed electrical signals along specialized cells called neurons. And these neurons secrete neurotransmitters, which are signaling molecules. And so we talked about this when we covered muscle tissue. I hope you remember that a motor neuron will transmit an action potential to a skeletal muscle cell, and the neurotransmitter at the neuromuscular junction is acetylcholine. So in the nervous system, communication actually happens via both electrical impulse, that's the action potentials, and chemical signals. Neurotransmitters are chemical signals that are released very locally between one cell and the next cell that is located in close proximity. The endocrine system, on the other hand, generally secretes hormones, these signaling molecules, that can travel in the blood, and our tissues respond more slowly to hormones, but the effects of the hormones tend to be much longer lasting. So both of these systems influence all sorts of aspects of homeostasis and growth, emotions, metabolism. So we'll spend a little bit more time talking about particularly where these 2 organ systems overlap. So the nervous system and the endocrine system are completely intertwined. Sometimes it's useful for us to speak about them separately. Sometimes it's useful for us to forget about the boundaries between the 2 and try to understand how the body works, where that category that we've created, this is endocrine and that's nervous. Sometimes that category doesn't really serve us because it's not an accurate reflection of reality. So a better term is the neuroendocrine system. So these are 2 examples of places in the body where the nervous system overlaps with the endocrine system. So in the panel on the left, where we're looking at number 1 and number 2, we are looking at a region of the brain called the hypothalamus and the pituitary gland. And nervous tissue makes up the nervous system. So nervous tissue includes, as we'll see, neurons and neuroglia. And all nervous system structures are made up of those 2 cell types, neurons and neuroglia. And we can see that there are neurons in the hypothalamus, and their axons extend down the infundibulum, that stalk between the hypothalamus and the pituitary gland. And those axons they extend into a portion of the pituitary gland called the posterior pituitary. So we tend to think of the pituitary gland as belonging to the endocrine system, but if that's the case, what are those neurons doing there? You know what those neurons are doing there? Those neurons release hormones. Particularly, they're called neurohormones, like oxytocin is a neurohormone that is secreted by those axons that extend from the hypothalamus into the posterior pituitary. So electrical signals directly cause neurons to secrete hormones like oxytocin into the blood. There's overlap there. And then the second panel on the right, number 3, we're looking at an adrenal gland there. And the adrenal gland sits on the superior aspect of each of your kidneys. So you have 2 adrenal glands. And at the core of the adrenal gland is called the adrenal medulla. And the adrenal medulla is made up of modified neurons that secrete the hormones epinephrine and norepinephrine. So we can see that there are the items that are labeled sympathetic preganglionic axons. We'll talk a lot more about what that means in this lecture topic. But what essentially happens in the adrenal medulla is modified neurons release hormones into the blood. So it's another example of where, can we tease apart the nervous system from the endocrine system? Not really. They overlap. So here's more vocabulary, because everybody needs more vocabulary in their lives. A neurohormone is a signaling molecule that was secreted by a neuron. So when we're talking about those cells in the posterior pituitary, or if we're talking about those cells that are in the adrenal medulla, those modified neurons, because they secrete hormones that circulate in our blood, those hormones are called neurohormones because they were secreted by a neuron. In this slide, we see 3 different mechanisms of cell-to-cell communication. And the panel all the way on the left represents the classic endocrine model. The panel all the way on the right represents the classic nervous system model. And the panel in the middle represents where those 2 systems intermingle. So let's just go left to right. We'll start with the classic endocrine model. In the endocrine system, classically, a gland secretes a hormone, and that hormone circulates in blood where it can bind to receptors on or inside target cells. In the middle panel, we see a neurosecretory cell secreting neurohormone. So that neurosecretory cell is a modified neuron, and the neurohormone, it's a hormone. It's a signaling molecule that was produced by a neuron. And that signaling molecule travels in blood and binds to receptors on target tissues. And then in the panel on the right, we see the classic nervous system model of communication, where neurons generate and transmit electrical signals, and those electrical signals, nerve signals, lead to the secretion of neurotransmitter molecules, and those neurotransmitter molecules bind to receptors on the postsynaptic cell. The structures that belong to the nervous system are all composed of what's known as nervous tissue. And that's one of the 4 basic categories of tissue in the body. Maybe you've heard this in another class. There is epithelial tissue, muscle tissue, connective tissue, and nervous tissue. We're going to focus on nervous tissue for a little while. So nervous tissue consists of 2 fundamental cell types, neurons and neuroglia. So let's think about those 2 fundamental cell types. Neurons are cells that are electrically excitable. I hope you remember that there are 2 cell types in the body that are electrically excitable, neurons and muscle cells. We're obviously going to focus on the neurons here in the nervous system. And then the second fundamental cell type that we find in nervous tissue is neuroglia. Neuroglial cells are not electrically excitable. They do not generate action potentials, and so they have that in common with the vast majority of cell types in your body. They maintain stable resting membrane potentials, just like most of the other cells in your body. And the neuroglia come in a variety of cell types that are all unique, and each one serves its own unique role in the nervous system. In one way or another, each type of neuroglia support the actions of neurons. Here we're looking at a sample of tissue taken from the central nervous system. It's a piece of spinal cord that was smushed, literally smeared across a slide. And the smearing process can separate the cells from one another so that it makes it easier for us to see those cells. After we stain the tissue, we add a cover slip, and then we use a light microscope. So we can see two fundamentally different types of cells here. The larger, obvious blue cell in the middle of the slide is the neuron. And neurons have a cell body. And the cell body is sort of like the central hub. The nucleus occurs in the cell body. Most of the cell's organelles occur in the cell body. And then there are radiating processes. A process is just something that juts off of something else. And so these processes are axons and dendrites, and we'll talk more about what axons and dendrites do in a little bit. So that's the neuron. Surrounding that neuron, all those tiny dark dots are the nuclei of glial cells or neuroglial cells. Sometimes they're just called glial cells for short. And glial cells are far, far, far more numerous than neurons. This is a great artistic representation for visualizing the different types of neuroglial cells as well as neurons. So what the artist has drawn here is a section of brain. So this is central nervous system tissue. And at the top, we see where you see it labeled cells of pia mater. Pia mater is the deepest layer of the meninges. There are cranial meninges around the brain and spinal meninges around the spinal cord. This happens to be brain, so that's the pia mater of the cranial meninges. And then we can identify the neurons. Neurons are shown in yellow here, and those are the electrically excitable cells. Those are the cells that generate and conduct action potentials. And then there are a variety of other cells in pink, in green, in blue. They have different sizes, different shapes, and they do different functions. Form follows function. And so, for example, the microglial cells, those act as immune system cells in the central nervous system. We see the oligodendrocytes in the central nervous system. Those oligodendrocytes create myelin sheaths, and we'll talk about the role of myelin. It wraps around the axons. We'll talk more about that a little bit later. We also have ependymal cells. So at the very bottom, that layer of pink cells, those are another type of glial cell called ependymal cells. And those ependymal cells are manufacturing cerebrospinal fluid, which is a fluid that bathes the exterior and some portions of the interior of your central nervous system. Your brain and your spinal cord are packed in cerebrospinal fluid. So it's not an exhaustive list. I don't need to talk about every single type of glial cell, but I would like you to know that glial cells are far more numerous than neurons. There are several different types of glial cells. We'll only focus on a couple down the road. And each type of glial cell plays its own role in maintaining homeostasis. Another reminder that muscle cells and neurons are the only two types of electrically excitable cells in the body. So what makes up your nervous system? It's a relatively simple design, but the function is oh so complicated. So humans, when we try to understand something, we often create categories, and often these categories are useful and sometimes they're less useful. This is a pretty useful category to separate structures of the nervous system into central nervous system or peripheral nervous system. So central nervous system, or CNS, it's shown there on the left. Your central nervous system consists of the brain and the spinal cord, and those two things are connected. You can think of your spinal cord as an extension of the brain, or maybe you care to think of your brain as an extension of your spinal cord. Both analogies, both ways of thinking about it work. And then on the right, we see structures that make up the peripheral nervous system. So anything that is made up of nervous tissue that occurs outside of the brain and spinal cord is considered part of your peripheral nervous system. And we see the basic structures there: cranial nerves, spinal nerves, ganglia, plexuses, and sensory receptors. So we'll talk a little bit more about each of these structures. This is just a different diagram to hopefully drive home the two basic boxes that we place the structures of the nervous system into: central nervous system or peripheral nervous system. So we'll talk more about each of these structures. Structure 1 and 2 here make up the central nervous system, and 3, 4, and 5 all occur outside the CNS and therefore belong to the PNS. So the human brain. The human brain is marvelously complicated. It is the most complex structure known in the universe. Of course, what is designating the human brain as the most complex structure in the known universe? The human brain is giving itself that accolade. It's pretty indisputable. No one has identified a more complex, sophisticated structure than the human brain. So your brain consists of roughly 86 billion neurons. No one ever counted. This is based on some estimates. And far more neuroglia than neurons. Your brain accounts for about 2% of your body weight, and it weighs roughly 3 pounds. Excuse me, your brain doesn't weigh a lot, but it does take up about 1/5 of your metabolism. So 20% of your resting metabolism is performed by the brain. So it weighs only 3 pounds, and think about how much you weigh versus your brain, and your brain gets 20% of the oxygen and glucose that are being delivered to your body tissues right now to support ATP synthesis. So one of the things that nervous tissue has in common with muscle tissue is that they are both metabolically demanding. They demand a high input of ingredients for supporting the continuous production of ATP because they use a lot of ATP to maintain homeostasis. And the brain is protected by the cranial meninges. So for part of this brain, the 2 layers of the meninges have been removed, the dura mater and the arachnoid mater, and the pia mater stays sort of adhered to the exterior surface of the brain. It covers all those folds and ridges. The spinal cord is the second component of your central nervous system, and it is continuous with your brain. There's no real discrete boundary between brain and spinal cord. One runs into the other. And your spinal cord, like your brain, your spinal cord is also encased in bone. It's protected by your vertebrae as it passes through the vertebral canal. And just like your brain, the spinal cord is covered in meninges, only we call them spinal meninges, but it has the same basic structure: the dura mater, the arachnoid mater, and the pia mater. Nerves. Nerves are part of the peripheral nervous system. They are bundles of axons traveling or coursing together. And nerves are bundles of axons that are surrounded by connective tissue. Here's a little aside. When I was in college and graduate school, I worked in a large hardware store, and eventually Home Depot put them out of business. But I worked there for 7 years, and I had a manager, and her name was Sharon, and she intimidated me. She intimidated everyone. She was not always a nice person to deal with, but she was hilarious, and for some reason she liked me. Um, and she would say to people, Joe, I've got one nerve left and you're on it. So now every time I think about nerves, I think about Sharon. She has since passed away. But maybe you have one nerve left and somebody's on it. All right, so here we are looking at nerves. There are cranial nerves and spinal nerves, and cranial nerves merge with or emerge from the brain, and spinal nerves merge with or emerge from, depending on your perspective, the spinal cord. So on the right, we can see the structure of a nerve, and it is bundles of axons. So these long electrical cords is one way to think about axons that course together, and these axons are, each one is coated in a layer of connective tissue. Connective tissue is not electrically excitable, and in fact, connective tissue acts as an insulator here, where if you think about each axon as a wire, and each wire in a nerve is insulated by the myelin sheath and the endoneurium. The endoneurium is a layer of connective tissue. And so if there is an action potential passing through one particular axon, that action potential will not excite neighboring axons because that endoneurium acts as an insulator. It's not electrically conductive. So this is sort of like coaxial cable. If you look at the end of a coaxial cable, you'll see little bits of wire, and each individual wire is coated in plastic. The metal wire will conduct electricity, the plastic does not, so it's an insulator. Ganglia. Ganglia are found in the peripheral nervous system, and they are masses of nervous tissue where we find the cell bodies of neurons. So we find neuronal axons in nerves, but for every axon there needs to be a cell body. Where are those cell bodies? Those cell bodies are either in the brain, the spinal cord, or in a ganglion. And so here we are looking at a particular set of ganglia called the sympathetic trunk ganglia. And they occur in 2 rows on either side of the vertebral column. You have ganglia other places in the body as well. And then finally, we have sensory receptors. You have a great variety of cells that are able to monitor changes in very different controlled conditions in your body. So sensory receptors are either free nerve endings, or they might include some type of specialized structure that is specialized to respond to changes in a particular type of physiological condition. So for example, we have cells that can respond to changes in stretch. We have cells that can respond to changes in temperature. We have cells that can respond to changes in pressure, etc. And so we have this huge variety of sensory receptors. And again, as humans, when we try to understand something that's complicated, step one often seems to be make categories. And again, sometimes these categories are really useful, and sometimes they are not useful. And furthermore, sometimes they get us into trouble when we try to oversimplify something that's really complex. So let's start with these categories. One way to classify sensory receptors is, well, do they respond to some change that occurs outside the body as part of our external environment, or do they respond to some change that's occurring as part of our internal environment? And so here are some examples of exteroceptors. And that just means that it's a type of sensory receptor that's paying attention to environmental stimuli on the outside of our body. So all of your touch receptors in your skin, and you have a great variety of skin receptors. Maybe you've studied them in another class and you remember things like Meissner's corpuscles, Pacinian corpuscles, the hair root plexus, things like that. So you have this variety of touch receptors in your skin that can detect temperature and pressure and vibration and an ant crawling on your arm. We also have olfactory receptors that are in the mucosa of your nasal cavity. The lining of your nasal cavity has these chemically sensitive cells where these cells are able to detect and respond to airborne molecules. So you can smell coffee and banana and chocolate. Light. In the retina of the eye, we have these light sensitive cells. So these are cells that will respond to, they generate signals in response to changes in light. And then another example here are our taste buds. We have chemically sensitive cells on our tongue, in our pharynx, even in our lungs, although we don't call them taste receptors in our lungs. But we do have these chemically sensitive cells that are able to respond to a variety of molecules that are dissolved in saliva so that you can taste coffee and chocolate and banana. And then here are just a few examples of interoceptors, sensory receptors that monitor factors, variables on the inside of our body. So we have previously talked about proprioceptors. Proprioceptors are responding to changes in muscle length, muscle position. Baroreceptors are stretch-sensitive cells that are monitoring your blood pressure. For example, we'll talk more about baroreceptors when we get to the cardiovascular system. And osmoreceptors, gosh, what do they monitor? Maybe they monitor the concentration of a body fluid. Ding, 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 ding. That would be correct. Osmoreceptors. And here the examples that are shown are in the hypothalamus as well as in the kidney. You have osmoreceptors other places as well. They are monitoring the chemical composition and the concentration of body fluids like blood, like cerebral spinal fluid, like urine that's in the process of being made. So previously I described the human brain as the most complex structure in the known universe. And whenever we want to understand something that's complex, we break it down into a much simpler form. And so now I'm going to tell you what might seem like the opposite. All of the functions that your nervous system is responsible for may be viewed through this model. There are three basic functions of the nervous system: the sensory function, the integration function, and the motor function. So here's a really simple model. Somebody sees a stimulus, right? Their photoreceptors are monitoring light. That's part of their external environment. Somebody sees a scary stimulus. Maybe somebody's coming at you and they're not wearing a mask and they're coughing, or maybe it's a bear. And so that's sensory input that travels along sensory neurons to the integration center. Sound familiar? Yeah, there's some overlap here with how homeostatic feedback mechanisms work. So sensory input travels along sensory neurons to the central nervous system. And in the central nervous system, there is an integration center. During integration, sensory input from multiple sources, usually, sensory input is combined, it's analyzed, and the central nervous system determines whether or not action is required. If action is required, the integration center gives rise to motor output. And those are electrical signals that travel along motor neurons to effectors. Effectors are either muscles or glands. So in this case, what did we see? We saw the scary bear. That stimulus was communicated to the integration center along sensory neurons. The integration center decided that that is worthwhile, worthy of a response. And so the integration center sent signals along motor neurons to skeletal muscle. Here it looks like we're looking at the gastrocnemius. And that gastrocnemius is now going to help you run away from the bear. So sensory input, integration, motor output. Very simple, right? We can apply this model to a lot of what your central nervous system does. I should say your nervous system, not just your central nervous system, your whole nervous system. So a little bit more about each of these. You have sensory receptors. Some of them are monitoring what's happening on the inside of your body. Others are monitoring what's happening on the outside of your body. Whatever type of sensory receptor we're talking about, it's monitoring some variable and it sends signals to the central nervous system. And those signals travel along afferent neurons. So this is the afferent communication pathway. Sound familiar? Yeah, you remember that word. Afferent neurons are also called sensory neurons. I love this image. This is a sagittal section through a human brain that's still connected to one eyeball, and that eyeball has also been sectioned. So where we see that number 1, it's sort of in the lower left region of the image. Where we see that number 1 is on the retina. So the retina includes photoreceptors, these light sensitive cells that generate electrical signals that travel along the yellow structure there between 1 and 2. That yellow structure is the optic nerve. So we see sensory receptors, we see a nerve, and as part of that nerve, there are sensory neurons. Afferent neurons are delivering information from the sensory receptor to the central nervous system. And at point number 2, that's the optic chiasm. That's where the 2 optic nerves from both eyes, the right eye and the left eye, meet the central nervous system, and they actually cross there. Do you see how it looks like a little X? Yeah. The optic chiasm is where input via the optic nerves enters the central nervous system, and those fibers cross each other at the chiasm. And so now we have sensory input that is in the central nervous system. So once we get past the yellow part in this image, that's the sensory input part, and we move on to the integration part. So in this image, where you see number 3 and number 4, those are processes that occur inside the central nervous system. And so they fall into the bucket called integration. So during integration, the central nervous system, the brain, or the spinal cord also can perform integration. The central nervous system typically receives sensory input from many different sources. So not only did you see the scary bear, but maybe you smelled the scary bear, maybe you heard the scary bear, and all of those sensory inputs travel to the integration center. And in the integration center, which is inside the central nervous system, those sensory inputs are analyzed, they're combined, they're weighed, which sensory inputs seem to be more important at that moment in time. And sometimes the integrative function involves neurons that are not sensory and they're not motor. And those are called interneurons. Let's use this image to reinforce what we've learned so far, and then we can expand on it. It's a really useful image for combining many different concepts. So overall, we're looking at a picture that demonstrates the mechanism behind the knee-jerk reflex. So the knee-jerk response is familiar to most of us. It's a common portion of a neurological exam. So is the nervous system functioning properly? And the patient is seated on an exam table where their feet can swing back and forth. So their feet are not resting on the floor, for example. And a little rubber mallet is used to gently tap the patellar tendon. And in response to that tap, the leg reflexively swings out. And this is what's known as a stretch reflex, and it is a protective mechanism. It's there to protect your muscles from being overstretched. In response to being overstretched, your muscles contract in order to counteract that stretch and prevent injury. So let's take a look at the sensory input, the integration, and the motor output. So sensory input in the knee-jerk stretch reflex arises with stretch receptors. These are proprioceptors. We talked about proprioceptors before. So these proprioceptors are embedded in the skeletal muscle tissue, and they are constantly monitoring the degree of stretch in your muscle cells, or between your muscle cells, really. How much stretch now? How about now? What is it now? And they're constantly sending signals to the central nervous system. And so when you bang on that patellar tendon, those cells stretch because they are connected to the tibia via the patellar tendon. And so when those cells detect an increase in stretch, they give rise to action potentials that travel along the sensory neuron into the spinal cord. And here we see the sensory neuron is represented in blue. We can call it an afferent neuron, we can call it a sensory neuron. Same. So once that message gets into the spinal cord, now the sensory function has ceased and the integrative function has begun. So now we're in the central nervous system. On the right, we see a section of spinal cord, and your spinal cord is part of your central nervous system. And if we follow that blue sensory neuron into the spinal cord, we're in the gray matter of the spinal cord, and we see that sensory neuron synapses on two different cell types. The green cell is an interneuron, and the red cell down below is a motor neuron. So that blue sensory neuron synapses communicates with both an interneuron and a motor neuron. So let's start with the interneuron. The interneuron shown here is green. It's neither sensory nor motor. And in this particular example, this is not always true for interneurons, but in this particular example, notice that the interneuron is connecting the sensory neuron to the motor neuron. So it is a pathway for communication between sensory input and motor output. The interneuron exists wholly, entirely inside the central nervous system. And let's continue thinking about that communication pathway from the sensory neuron to the interneuron to that particular motor neuron, the lower motor neuron, the bottom motor neuron in this image. So following the message that, hey, the quadriceps have just stretched, that sensory neuron will synapse and deliver information to the interneuron in green. That interneuron will then fire an action potential and secrete neurotransmitter at the synapse with the motor neuron. That neurotransmitter will serve to prevent an action potential in that bottom motor neuron. And let's look at what that motor neuron connects with. That motor neuron, this bottom motor neuron here, this one connects with the hamstrings, right? And during the knee-jerk reflex, the hamstrings need to stay relaxed. So by preventing an action potential in that motor neuron, the interneuron helps to keep the hamstrings relaxed, not contracting. And this will enable the quadriceps to actually cause extension of the leg at the knee. All right, let's look at the second synapse shown here between the sensory neuron, and this time there's no interneuron involved, it just synapses directly with a motor neuron. So when during integration, this sensory input that's delivered from the sensory neuron causes communication between a sensory neuron and a motor neuron, or an afferent neuron and an efferent neuron. And in this case, the neurotransmitter that gets secreted at the synapse excites this motor neuron, and this motor neuron will send an action potential. And here we are at the neuromuscular junction in one of the muscles of the quadricep. And you know what happens here. You know that the action potential in the motor neuron leads to the secretion of acetylcholine. You know that acetylcholine binds to acetylcholine receptors at the neuromuscular junction and causes ligand-gated sodium ion channels to open up, the muscle cells depolarize, and that will lead to muscle contraction. The muscle contraction in the quadricep actually causes this motion right here, or this action. This is called flexion of the leg at the knee. So, motor output. Motor output arises in the integration center and it travels along motor neurons, which are also known as efferent neurons, from the CNS to the effector. And effectors are always muscle cells or gland cells. And then the muscle cells or the gland cells are able to respond to the signals sent by the integration center. So you remember this, our good friend, the neuromuscular junction. Now we're pretending that to go along with the knee-jerk reflex, now we'll pretend that these are skeletal muscle cells in, let's say, the rectus femoris, which is one of the muscles that makes up the quadriceps, and an action potential will travel down to these axon terminals, to these synaptic end bulbs, and acetylcholine will be released and these muscle cells will contract. So you're most familiar with skeletal muscle cells at this point, acting as effectors, but cardiac muscle cells are effectors, gland cells are effectors, smooth muscle cells are effectors. So pick a motor neuron, any motor neuron, and it's going to deliver signals to any of these: skeletal muscle, smooth muscle, cardiac muscle, or glands. So here are two examples of glands that are controlled by motor neurons. So you have those salivary glands. You have 3 sets of salivary glands, and they produce saliva. You have your parietal gland, which is sort of anterior to your ear on both sides. And then you have your submandibular gland, which is below your mandible. And then you have your sublingual glands, which are below your tongue. And so in response to, let's say you smell something delicious and you start to salivate, there was sensory input, integration, and motor output. And your salivary gland cells responded to action potentials delivered by motor neurons, and they responded by producing more saliva. And then you have a variety of skin glands. The ones that we tend to be most familiar with are the eccrine sweat glands. Here, they're just sweat glands. So the temperature in the room goes up, right? That's detected by those thermoreceptors that are present in your skin and mucous membranes, and they send their sensory input to the site of integration, which happens to be in the hypothalamus, and the hypothalamus gives rise to motor output. In this case, we're talking about sweating. And so that motor output signals the cells that make up eccrine sweat gland to release the components of sweat, which will then travel through this duct and onto the surface of your skin. And that sweat will help cool your body by evaporation. We can summarize the sensory, integrative, and motor pathways by looking at this image. I always make up that this gentleman is writing a Dear John letter. Do you know what a Dear John letter is? A Dear John letter is a breakup letter. You write the letter and you leave it on the kitchen table or on the mantle, and it's like, Dear John, I'm leaving you. And then you pack up your stuff and go. So this guy is breaking up with his boyfriend. And can we trace the sensory, integrative, and motor pathways? And so I asked the class, what color did the artist use to represent the sensory pathway? What color? Blue. We see the sensory receptor. I don't know, maybe that's a Meissner's corpuscle in his index finger, and there's a sensory neuron traveling up his arm toward the spinal cord. And once we get into the spinal cord, we move from the sensory to the integrative. And the next question I asked the class is, what color did the artist use to represent interneurons as part of the integrative pathway? And those interneurons are represented sort of in like a purplish gray color. As we get up through the spinal cord and into the brain, those purplish gray neurons are interneurons. They are neither sensory nor motor. And the next question I asked the class is, what color did the artist choose to represent the motor pathway, the efferent pathway? And that's that dark red color. And so we see the motor signals arise in the central nervous system, in the primary motor cortex, that's part of your brain, we'll talk about it later. as you're thinking about what to write, and you need to generate the muscle movement in order to form the letters D-E-A-R, dear, John. Those signals arise in your primary motor cortex in your brain. They travel as part of the motor pathway down your spinal cord, out through a spinal nerve, and to the muscles in your arm and your hand that are required for you to write. So we have sensory, integration, and motor pathways. Up next, we're going to discuss the organization of the nervous system in terms of which components talk to which sensory receptors and which effectors. So this is organization of the nervous system in terms of who controls what. There are many ways to depict the organizational framework of the nervous system. This is the most simple diagram that I have for you today, where the nervous system is fundamentally divided up into central nervous system, the brain and spinal cord, and the peripheral nervous system. And we divide the peripheral nervous system into 3 divisions: the somatic nervous system, the autonomic nervous system, and the enteric nervous system. We will spend most of our time this semester talking about the somatic nervous system and the autonomic nervous system. The enteric nervous system, enteric means intestine. The enteric nervous system largely governs functions that occur as part of the digestive system, all the muscular action and the secretion of various enzymes and juices. And we're going to mostly ignore the enteric nervous system for now, and we'll talk about it more when we cover the digestive system. So in the nervous system lecture topic, we'll focus mostly on the somatic nervous system and the autonomic nervous system. We'll keep the enteric nervous system in mind, but I'm not going to tell you very much about it yet. This is obviously a more complex representation of the divisions of the nervous system, and the arrows indicate the flow of information. So up top, we have the central nervous system, and then down below, sort of in that large purple rectangle, we see those 3 branches in the nervous system that I just talked about: the somatic nervous system, the autonomic nervous system, and the enteric nervous system. This is a different representation of the same type of information. It's often helpful for learning to have multiple views of the same type of information. So let's focus on the similarities and differences between the somatic nervous system and the autonomic nervous system, the SNS versus the ANS. So the somatic nervous system is responsible for consciously perceived sensations arising from your skin, your joints, and your skeletal muscles. And generally, motor output is under voluntary control. So soma means body. So these are the bodily sensations that you have awareness of. You can talk about them. You could point to, part of your leg that hurts. You can tell the healthcare provider. And motor output is generally under voluntary control because the effectors here are skeletal muscle. And we generally have voluntary control over skeletal muscle. So here we can take a more detailed look at the jurisdiction of the somatic nervous system. So we can talk about that in terms of, well, what types of sensory input does the somatic nervous system handle, and what types of motor output does the somatic nervous system generate? And in a moment, we're going to contrast this with the autonomic nervous system. But the somatic nervous system deals with sensory input that arises from your 5 senses. So hearing, sight, taste, smell, touch. Proprioceptors, which we've talked about several times now, those are sensory receptors in your muscles and your joints, and they give you a sense of your body position in space. And then your vestibular system, which is responsible for your sense of balance and equilibrium. So sensory input arising from only those types of sensory receptors feed into your somatic nervous system. And then, of course, integration occurs in the central nervous system, either in the brain or the spinal cord. And the CNS can generate motor output. In the somatic nervous system, the type of motor output that is generated only goes to skeletal muscle. So smooth muscle is not part of the somatic nervous system, cardiac muscle, not part of it, glands, not part of it. The only effector in the somatic nervous system is skeletal muscle. And in the domain or the jurisdiction of the somatic nervous system, we generally have a lot of conscious awareness over these sensations that arise from your five senses. Obviously, you're aware of what you see, what you smell, what you hear. You have a sense of balance. You know where your body is in space. And motor output is usually under voluntary control. You can willfully stand up right now and go and get a glass of water. And an important contrast with the autonomic nervous system is motor output can only stimulate the effector. So anytime a somatic motor neuron sends a nerve impulse, that will cause skeletal muscle to be excited, and excited means generates an action potential. So that skeletal muscle, as you know, will contract in response to motor output in the somatic nervous system. And then in that image there, we are looking at a communication pathway where the sensory input is arising from some type of touch receptor in the skin. And that sensory information travels along a sensory neuron, also known as an afferent neuron. And integration in this case is occurring in the spinal cord. And in that drawing, we can see that the sensory neuron is synapsing with a motor neuron. And this is a somatic motor neuron. And that somatic motor neuron delivers efferent signals to skeletal muscle. So that effector there is going to be skeletal muscle. And when this pathway is engaged, you generally have conscious awareness of it. You can feel what's happening in your skin, and you know that your skeletal muscle is contracting in response. Up next, we're going to contrast the somatic nervous system with the autonomic nervous system. So the autonomic nervous system, the ANS, is responsible for subconsciously perceived sensations arising from your visceral organs, your internal organs. And generally, motor output is not under voluntary control, and we often don't have great conscious awareness over it. So let's break that down into sources of sensory input and what types of effectors are part of the autonomic nervous system. So sensory input arises from interoceptors, the variety of sensory receptors that are paying attention to the various forms of physiological variables that are happening inside your body. Chemoreceptors, baroreceptors, osmoreceptors, etc. And integration is going to occur in the brain, and motor output, the brain will generate motor output that travels along motor neurons, autonomic motor neurons. And in the ANS, the effector might be smooth muscle cells, it might be cardiac muscle cells, and it might be glands. It is not skeletal muscle. And so this is what I like to think about as your zombie brain. There's a lot of your brain is dedicated to controlling things that you have very little conscious awareness over. So your brain really does have a mind of its own, and it is governing all sorts of aspects of homeostasis below your level of awareness. So for example, do you have great awareness over exactly how much urine is in your urinary bladder right now? Do you really know what your blood pressure is? Can you feel that right now? I hope you're nice and calm. And the answer to that is no. And motor output is generally under involuntary control. So things are happening where you don't need to think about it. So if you stand up right now and go and get a glass of water, you know what's going to happen to your heart rate? Your heart rate's going to go up a little bit, and your heart is going to contract a little harder, and that will raise your blood pressure a little bit, and that will allow your blood to circulate faster to deliver oxygen and nutrients to the skeletal muscles that are helping you go and get a glass of water. Now, you know about the skeletal muscles. You know that you're moving through the room because that's the somatic nervous system. The autonomic nervous system is in charge of increasing your heart rate and your blood pressure, and you have little awareness over that. Motor output is divided into two different categories in the autonomic nervous system. Motor output either flows along sympathetic motor neurons or parasympathetic motor neurons. And so for those internal organs that we see in the diagram there, like we see some airways in the lungs, we see the heart, that's a stomach and a duodenum, the first region of the small intestine. And then there's a pancreas below that and an adrenal gland. And then we see part of the large intestine. And I think that's a scrotum and a penis and the urinary bladder. So those are all organs that include smooth muscle or cardiac muscle or glands, and they receive two different types of motor output from the ANS. They have what's known as dual innervation. So let's take a look at that heart. Do you see how there's two lines traveling from the spinal cord toward the heart? One is a solid line, one is a dotted line. The artist drew that in there to represent dual innervation. One of those motor neurons is a sympathetic motor neuron, and it delivers sympathetic stimulation to the heart. The other motor neuron, maybe it's the dotted line, it is a parasympathetic motor neuron, and it delivers parasympathetic stimulation to the heart. One will increase the activity of the effector. The other will decrease the activity of the effector. So if we're thinking about the heart, sympathetic motor output increases heart rate and increases the force with which the heart contracts. The opposite of that, it will be caused by parasympathetic motor output. So the parasympathetic motor neuron will deliver stimulation to the heart, only it has an inhibitory effect. It will help slow the heartbeats. So after you get that glass of water and you sit back down and you sink into this nice, comfortable lecture, your heart rate will go down because your heart received an increase in parasympathetic motor output. Here's a side-by-side or top and bottom comparison of motor output in the somatic nervous system, that's up top, and motor output in the autonomic nervous system. So let's start with the simpler system. The somatic nervous system is more simple than the autonomic nervous system. So let's start with the simple and then move on to the more sophisticated. So let's start with the top panel where we see a section of spinal cord and we see a green motor neuron. Notice that the cell body of that motor neuron is in the gray matter of the spinal cord. Notice that the axon of that motor neuron exits the spinal cord through a spinal nerve, and then it travels as part of a nerve to the effector. Notice that the effector here is skeletal muscle cells, and you recognize ACH. That's acetylcholine. So every time that green somatic motor neuron fires an action potential, that action potential results in the secretion of acetylcholine, and acetylcholine will stimulate the muscle cells and cause muscle contraction. Notice that there is just one motor neuron that delivers an action potential from the spinal cord to the effector, to the skeletal muscle cells. Okay, let's go to the bottom panel and look at autonomic motor output. So let's start with our section of the spinal cord. Again, we have a green motor neuron and its cell body is in the gray matter of the spinal cord. That's the lateral gray horn of the spinal cord. And let's follow that green motor neuron. Let's follow its axon. Its axon exits the spinal cord as part of a spinal nerve. And then that motor neuron travels as part of a nerve to a ganglion. So we're looking at the presynaptic motor neuron, or sometimes we call it the preganglionic motor neuron, or sometimes it's just easier to say motor neuron number 1, although that is not the technical term. So there are two motor neurons in the autonomic efferent pathway. So let's follow motor neuron number 1. That's our preganglionic or our presynaptic motor neuron. And that is going to secrete acetylcholine in an autonomic ganglion where the synapse occurs with motor neuron number 2, the postganglionic motor neuron, also known as the postsynaptic cell. So motor neuron number 2 begins in the ganglion and then travels to the effector. In this case, the effector is cardiac muscle tissue. And this pathway of motor output is either sympathetic or parasympathetic. This diagram doesn't designate it as one or the other, but this diagram allows us to envision both possibilities. In this diagram, if this postganglionic motor neuron secretes acetylcholine, then we know that it is a parasympathetic motor neuron. Motor neuron number 2 in the parasympathetic pathway secretes acetylcholine, and acetylcholine, because we're looking at cardiac muscle tissue, acetylcholine will slow down heart rate. Now let's imagine the other scenario. This could be a sympathetic motor pathway. And so now let's imagine that motor neuron number 1 and motor neuron number 2 are sympathetic motor neurons, sympathetic efferent neurons. In the sympathetic case, the neurotransmitter that is secreted at the junction between motor neuron number 2 and the effector, cardiac muscle tissue in this case, would be norepinephrine. That's what the NE stands for. And norepinephrine will cause cardiac muscle cells to will cause the heart rate to increase and will cause the heart to beat with greater force. So, to recap or to point out the most salient differences between the somatic and the autonomic motor output. Let's start and review the somatic. In the somatic nervous system, motor output occurs along a single motor neuron. One motor neuron runs from the central nervous system to the effector, and the effector is always skeletal muscle. Acetylcholine is always released by the motor neuron, and acetylcholine, you know, stimulates skeletal muscle cells. So skeletal muscle cells will generate action potentials. Okay, let's contrast that with what's going on below in autonomic motor output. There's a 2 motor neuron pathway. So the first motor neuron, the cell body resides in the central nervous system, in the spinal cord or in the brain. And motor neuron number 1 synapses with motor neuron number 2 inside an autonomic ganglion. And then motor neuron number 2 will release a neurotransmitter onto the effector. And the effector is either cardiac muscle, smooth muscle, or gland. And the autonomic motor output is either sympathetic or parasympathetic. One form of stimulation along one pathway will cause whatever the effector is, it will cause it to increase its activity. And stimulation occurring along the other pathway will cause the effector to decrease its activity, will inhibit the effector. And we will talk about many examples of this down the road. Here's an easy quiz. So input and output. For each of the questions below, determine which branch of the peripheral nervous system is most responsible. So number 1, you feel an itch on your arm and you scratch it. Is that SNS or ANS? It's the SNS, right? You feel an itch on your arm. You have great conscious awareness over it. The sensory input is arising from your skin, and then you scratch it. That's motor output that involves skeletal muscle. It's voluntary, you know what's happening. Number 2, your body temperature starts to rise and your sweat glands begin to secrete sweat onto the surface of your skin. Is that SNS or ANS? It's ANS. Your body temperature starts to rise, and that's detected by interoceptors, those thermoreceptors that are present in your skin. They're also present in your mucous membranes as well as in your brain. And those sweat glands, that's glands. So the effector, one of the effectors in the autonomic nervous system are glands. And This happens at a subconscious level. You can start to sweat, and you don't even have to get sweaty to be sweating. Your sweat glands can start to release sweat onto the surface of your skin, and you have no conscious awareness of it. And it evaporates before it ever collects as droplets of water. You have no knowledge of this. So this is clearly autonomic nervous system. Number 3, the lights in the room suddenly go out and your pupils dilate. Somatic nervous system or autonomic nervous system? Autonomic nervous system. So there is smooth muscle in the iris of your eye that's responsible for the diameter, the size of your pupils. And smooth muscle is under the jurisdiction of the autonomic nervous system. And then number 4, you think about your crush or your sweetie and your heart rate increases. Is that caused by your somatic or your autonomic nervous system? It's your autonomic nervous system. And specifically, there'll be an increase in sympathetic stimulation to your heart, and your heart rate will go up. And then there are the answers if you ever need a reminder. All right, our next topic is to learn more about the structure and function of neurons. So here we get to apply some of the knowledge that we acquired during our coverage of cell-to-cell signaling. So we will be reviewing action potentials and then building on that. There are always, in biology, there are always deeper layers. So here's the next level of depth. So this is a super simple diagram of a neuron. And neurons, in reality, they come in many different forms. They have different morphologies. They have different shapes. In physiology, we are concerned with understanding how all neurons work, or the general neuron. And so it's fine for us to just use this particular shape of a neuron as our example. But if you pursue studies, like if you ever take a neuroscience course, get ready to learn about different neuron morphology. So they have different shapes. They all work basically the same. So all neurons have a cell body, which is sometimes called the soma. And in that cell body, we find the nucleus, and we find most of the other organelles, the endoplasmic reticulum, most of the mitochondria, etc. And then connected to that soma, or branching from that soma is perhaps a better way to say it, are neuronal processes. A process is anything that juts off something else. And so there are 2 types of neuronal processes. There are dendrites and there are axons. And this neuron, and the type of neuron that we'll always be talking about in this class, has just a single axon, and there are many dendrites. And the dendrites are for receiving signals from neighboring cells, and the axon is for transmitting or sending signals to neighboring cells. This axon happens to be myelinated. That's not true of all axons. This one happens to be myelinated, and we'll talk more about the structure and the function of myelin in a little bit. In general, myelin serves as an insulator, and it helps action potentials travel faster. And we'll talk about that mechanism, how that occurs, a little bit later. One more structure that I need to point out here is the junction between the cell body, or the soma, and the axon. Where the axon and the cell body meet is a region of the neuron called the axon hillock. And it's also sometimes called the trigger zone, because as we will see, this trigger zone is important for determining whether or not this axon will generate and transmit an action potential. So here's some vocabulary in case you need to refer back to it. I just covered all of these terms in the previous slide. And here we see 2 neurons connected in sequence. We've looked at this picture before when I introduced the concept of synapse in the cell-to-cell signaling lecture topic. And so we have the presynaptic cell and the postsynaptic cell. You can also think about this as the talking cell and the listening cell, or neuron number 1 and neuron number 2. But the technical terms here are really the presynaptic cell and the postsynaptic cell. And what I'd like to draw your attention to next are all the red arrows. So just let your eye pass over that image and look at all the red arrows. Not so much the zoom in the lower left. Don't look at the synapse quite yet. But we do see red arrows in the dendrites of the presynaptic cell. And those red arrows are indicating that this cell is receiving stimulation from other neighboring cells, not shown here. So that stimulation generally comes in the form of neurotransmitters that bind to receptors. And if the right neurotransmitter is secreted by some neighboring cell, it will cause the dendrites and the cell body, the plasma membrane along the dendrites, covering the dendrites and the cell body, it will cause that plasma membrane to depolarize. And if that wave of depolarization brings the axon hillock to threshold, negative 55 millivolts, then an action potential will be generated and propagated. It will travel the length of the axon in that presynaptic cell. And now we get to look at the zoom in, right? This is a chemical synapse. We've seen it before, where the presynaptic cell will release neurotransmitter into the synaptic cleft. This is just labeled neurotransmitter. There's more than 100 different neurotransmitters in your body. So far, we've talked about acetylcholine. We will go on to talk about norepinephrine. Those are the two neurotransmitters that we'll spend most of our time talking about. There are many, many others that exist. So this neurotransmitter is a chemical signal that diffuses across the synaptic cleft and then goes on to bind to receptors in the plasma membrane of the postsynaptic cell. So now we're moving on to neuron number 2. The postsynaptic cell, neuron number 2, in response to neurotransmitter that was secreted by the presynaptic cell, the postsynaptic cell, the plasma membrane covering its dendrites and its cell body might depolarize, might go from negative 70 millivolts to negative 55 millivolts, and negative 55 millivolts is the threshold. And so at that axon hillock, an action potential will be generated, and it will travel along the entire length of the axon of the postsynaptic cell, all the way to the axon terminals and those little knobbly synaptic end bulbs, where we just see those two red arrows all the way on the right, indicating like, hey, this is where the signal gets transmitted to another cell, but that other cell is not shown in the picture. I love scanning electron micrographs. So here we can see, this is just a tangle of neuronal processes and some neuroglia and the cell body of one neuron. And I love that tangle of neuronal processes. So those are axons and dendrites from neighboring neurons, and they're all crisscrossing. And then the little green blobs are neuroglial cells. The central nervous system is super duper complex because of the way neurons are connected to each other. Which neurons talk to which other neurons? It's very complex. It's often likened to the internet, this web of connection. And because it is so complicated, it is difficult to understand. And we are far from a complete understanding of the nervous system. We don't understand the physiological basis of consciousness. Why are you aware of the fact that number one, you exist? Why do you have that awareness? Why do you have a sense of self? Like you exist. Do you think an oak tree has a sense of self? Well, we have no way to know, but there's no evidence to suggest that it does. So these, really compelling questions in neuroscience remain, a lot of them. And the nervous system is difficult to study because it is so complicated. So it takes a lot of time and a lot of different research groups working all over the world to hopefully one day answer even some of these basic questions.
All right, ready for our next level of complexity? We have talked about action potentials. There is another type of electrical signal called a graded potential. And we will compare and contrast graded potentials with action potentials. So graded potentials are for short distance communication only. They occur along the plasma membrane that's covering those dendrites and the cell body of a neuron, but that's it. They die off. And graded potentials can vary in amplitude. And I'll show you that on a graph in just a few minutes. But it's possible to have a small graded potential or a large graded potential, depending on the stimulus. In contrast, action potentials arise in axons and are used for both short and long distance communications. So we have lots of neurons in our brain that have these tiny little axons, and an action potential might travel a millimeter. And we also have these neurons that have these really long axons. So if you can wiggle your toes right now, the motor neuron cell body that sends those action potentials to the skeletal muscles in your toes that allow your toes to wiggle, the cell bodies of those motor neurons are in your spinal cord. And the axon extends from your spinal cord all the way down to your toes. So that's a pretty darn long cell. And imagine, a professional basketball player. And then action potentials, they are all one size and they are all or nothing. We will review that graph of an action potential, and for a neuron, an action potential will always look like that. Depolarization will happen, repolarization will happen, hyperpolarization happens in some neurons and not others, and then the cell returns to resting membrane potential. There's no such thing as a small action potential, no such thing as a big action potential. They're one size and they're all or nothing. They happen or they don't. It's full on or full off. This is a great diagram, and obviously we're looking at a single neuron here, and the plasma membrane of this neuron is color-coded. So we're looking at the entirety of just a single cell here, and the dendrites and the soma, the cell body, the dendrites and the cell body of this motor neuron, the plasma membrane covering those parts of the cell, are coded in blue. And this is because we need to see where graded potentials are possible. So graded potentials arise in dendrites or on the cell body. And these graded potentials travel, they propagate for short distances, and then they die out. Graded potentials can give rise to action potentials. And notice the green part of the plasma membrane of this neuron. The green highlighted part includes the axon and the axon terminals, those branches down at the bottom, and the synaptic end bulbs in red. So an action potential, if it arises, it will arise at the axon hillock, or the trigger zone, where that axon meets the cell body. So where blue plasma membrane meets green plasma membrane, that's the axon hillock or the trigger zone. And if an action potential occurs, then that action potential will definitely travel the entire length of that axon all the way to the synaptic end bulbs. And if it's a chemical synapse, then neurotransmitter will be released. Once that action potential starts, it's full on, it's all. It's not going to start and then die out. And there's no such thing as a big action potential or a small action potential. All right, so let's talk more about graded potentials. So a graded potential, so we're talking about electrical signals that arise in the plasma membrane around dendrites or the cell body. What happens is there's a change in membrane potential that's caused by a certain set of ion channels opening up. And that set of ion channels opens and a particular ion either enters or leaves the cell according to its electrochemical gradient. And ions are charged, as you know, and that moving ion entering the cell or leaving the cell, depending, is going to take its charge with it. So the inside of the cell could become more positive, or the inside of the cell could become less positive, more negative. Either way, the membrane potential will change, and that's called a graded potential. So we need to do a little bit of conceptual review in order to fully understand graded potentials and action potentials. So I hope you remember that physiological electric signals are caused by the rapid movement of ions across the plasma membrane. The flow of charged particles is electricity, and in physiology, those charged particles are ions. And this is going to cause temporary changes in the voltage difference across the plasma membrane, the membrane potential. And we'll review what happens to membrane potential, how does it change numerically and directionally during hyperpolarization and depolarization. Oh, you remember this picture. This picture we looked at in the cell signaling lecture topic, and in this image we see a phospholipid bilayer that's meant to represent the plasma membrane. And we see the distribution of various charged particles outside the cell versus inside the cell. And I'll remind you that outside the cell, we find relatively higher concentrations of chloride and sodium. And inside the cell, we find relatively higher concentrations of potassium, negatively charged proteins, and negatively charged phosphate groups. So for each of these solutes, chloride, sodium, potassium, they all will move, given the opportunity, along their respective electrochemical gradients. And let me also take a moment to remind you that the phosphates and the proteins are trapped inside the cell. There's no way for them to get across the plasma membrane. So we're really talking about the movement of the ions here. Chloride, sodium, and potassium, they could all move across the membrane as long as an ion channel was present and open. So now let's play that game again where you imagine that there are potassium ion channels present in this membrane. Put them in there and now open them. Potassium ion channels are there and they're open. In which direction will potassium flow? Will more potassium enter the cell or leave the cell? Think about the electrochemical gradient. More potassium will leave the cell. That's right. And what's going to happen to membrane potential? Think about the inside of the cell. This cell has a resting membrane potential of negative 70 millivolts. And you just said a whole bunch of potassium is going to leave the cell and take its positive charges with it. What's going to happen to the inside of the cell when the cell loses a bunch of positive charges? The inside of the cell is going to become more negative relative to the outside of the cell. So the membrane potential will go from negative 70 to negative 75, negative 76, negative 80. It depends on how long those potassium ion channels stay open, but this membrane will hyperpolarize. We've seen a graph like this before. Let me remind you of what's on the axes. On the vertical axis, we have membrane potential in millivolts, and on the horizontal axis, we have time in milliseconds. And this cell, our standard neuron, has a resting membrane potential of negative 70 millivolts. And resting means that the cell is not generating physiological electric currents. It's just holding steady at negative 70 millivolts. And then some stimulus comes along to cause the cell to hyperpolarize. Do you see how we go from negative 70 to, I don't know, maybe that's negative 74. And then those ion channels close. Maybe they're potassium ion channels. Potassium ion channels close, and once again, those sodium-potassium pumps are able to restore resting membrane potential. So this is a brief change in membrane potential. It is a graded potential. It is a hyperpolarizing graded potential because the inside of the cell got further away from zero. The inside of the cell became more negative relative to the outside of the cell, and then it died out, and the cell went back to resting membrane potential. All right, back to this exercise. Now, I would like you to imagine that there are sodium ion channels in this membrane and open them. In which direction will more sodium ions flow? Will more sodium ions leave the cell or enter the cell? That's right. More sodium ions will enter the cell because sodium ions will flow by facilitated diffusion along their electrochemical gradient. What's going to happen to membrane potential? When sodium ions enter the cell, they bring all of their positive charge with them. What happens to the inside of the cell? Does the inside of the cell get more negative or less negative? We start at -70. A bunch of sodium is going to enter the cell. What happens to membrane potential? Does it become more negative or less negative? It becomes less negative. As a whole bunch of positively charged particles enter the cell, the inside of the cell becomes less negative relative to the outside of the cell. So that cell that started off at -70 is going to maybe go -69, -65, maybe it'll hit -60. So that we'll see is called a depolarizing graded potential. And here's the graph. You recognize the axes. You recognize resting membrane potential. Something causes sodium ion channels to open up. A whole bunch of sodium enters the cell and membrane potential deviates from -70 all the way to, oh, it looks like that's about -60. And then those ion channels close and the sodium potassium pumps restore the membrane potential to resting. This is known as a depolarizing graded potential. So the graded potentials that are generated in the blue parts of the plasma membrane, in the dendrites and the cell body, those graded potentials, depolarizing, hyperpolarizing, they will determine what happens at the axon hillock, at the trigger zone. If that trigger zone reaches the threshold of -55 millivolts, then in the green parts of the plasma membrane, voltage-gated sodium ion channels will open up, and bam, we have the depolarization phase of an action potential. Speaking of action potentials, you recognize this graph, I hope. This is the graph of an action potential in a typical neuron, and once we reach that threshold of -55, the voltage-gated sodium ion channels will open, giving rise to B, the depolarization phase of an action potential. Now that black arrow on the graph is indicating a depolarizing graded potential. Yeah, I didn't tell you that before, but we're adding the next layer of complexity. So as this cell goes from A to B, A, the cell is at resting membrane potential. As the cell is stimulated because some set of ion channels open up, causing a depolarizing graded potential, the cell goes from -70 to -65 to -55. And as soon as we hit -55, that depolarizing graded potential is now over and it has initiated an action potential. I hope you remember this diagram as well. We can use it to review the events that occur during an action potential. We can start with a cell that's at resting membrane potential. So we'll start with number 1 at the top. And sodium ion channels are actually pretty sophisticated. And notice in that top panel, in the green sodium ion channel, that we see labels are activation gate closed, inactivation gate open. That's true and correct information. I'm not going to go into that level of detail. For us, these voltage-gated sodium ion channels will be either open, closed, or resting. And I'll talk more about that in a little bit. So let's just review what we know about action potential, starting with number one, the neuron is at rest. It's not generating any electrical signals. There are no graded potentials. There are no action potentials. The cell is at resting membrane potential. What's going on? Voltage-gated sodium ion channels are closed and voltage-gated potassium ion channels are closed. Moving from number one to number two, when we enter the depolarization phase of an action potential, some stimulus brought membrane potential to the threshold of -55, and now we know that stimulus was a depolarizing graded potential. Got the membrane potential at the axon hillock to -55. At -55, voltage-gated sodium ion channels open up, and sodium rapidly enters the cell, bringing about that super dramatic depolarization phase of an action potential as membrane potential moves from -70 millivolts all the way up to +30 millivolts. And then we move from step 2 to step 3, the repolarization phase. As we approach +30 millivolts, the sodium ion channels close, and now they're resting. And the potassium ion channels open up, and there's this massive exodus, outflow of potassium from the cell, and that causes the inside of the cell to become increasingly negative, and we get that red portion of the graph called the repolarization phase. So moving from step 3 to step 4, repolarization continues. Hyperpolarization might happen depending on the neuron. It's not represented in this action potential, and that's okay. Not all neurons exhibit a hyperpolarization phase. So we can finish repolarization, and the sodium-potassium pumps will restore resting membrane potential. Those voltage-gated sodium ion channels are closed. Those voltage-gated potassium ion channels are closed. And we return back to number one, resting membrane potential. This is a great diagram. I love when things are color-coded. So let's start up top. We see our neuron, we see the dendrites, we see the cell body, we see the axon is highlighted in green and all the axon terminals and the little synaptic end bulbs. And so it's highlighted in green to remind us that action potentials only occur in the axon and the axon terminals and the synaptic end bulbs. Notice that the dendrites and the cell body are not highlighted in green because graded potentials occur there. And notice that the graph below is an action potential. So here we get to look at the different phases of the action potential and use this color coding to remind us of what's happening, and we can add more layers of sophistication. So let's start on the graph where the cell is at resting membrane potential, and there's that little arrow that says stimulus. This stimulus is occurring somewhere along the dendrites and or the cell body, and it causes a set of ion channels to open that just locally change the membrane potential. And if the membrane potential at that axon hillock hits -55, then the voltage-gated ion channels, sodium ion channels, fly open, and we get depolarization and repolarization. And in this slide, this neuron, we have a hyperpolarization phase and so on. So let's take a look at this color coding. In sort of that, they're very Eastery or spring colors, I guess, that top lavender color. In this portion of the graph, wherever you see that lavender color, the cell is exhibiting resting membrane potential, and the voltage-gated sodium channels are closed and the voltage-gated potassium channels are closed. There is no physiological electric current, and the cell is at -70. In that pretty narrow light blue phase, that's a depolarizing graded potential that brings the axon hillock to threshold. And then we get sort of like that light green gray color. That's the depolarization phase, followed by the pink color. That's our repolarization phase. And that sort of orangey tan color is the hyperpolarization phase. And then we return to resting membrane potential. All right, so if we look all the way to the right there, where we see those brackets that indicate something called the absolute refractory period and the relative refractory period. So to be refractory means that another stimulation cannot occur. So in this context, it means that another action potential cannot occur when a neuron is in the refractory period. There are two levels of being refractory here, absolute and relative. During the absolute refractory period, another action potential is not physically possible. It's not physiologically possible. It cannot occur. And I'll tell you why, but let's look at the absolute refractory period. So we have that sort of light grayish green and that light pink color. So look at what's going on. The absolute refractory period corresponds with the depolarization phase of an action potential and the repolarization phase of an action potential. Let me explain that absolute refractory period to you. Why can't another action potential occur during these two phases of an action potential? Well, number one, an action potential is already happening. So the voltage-gated sodium ion channels are already open. There's no way that we can open them again. They're already open. So we can't, there's just no way to have another action potential on top of the already occurring action potential. And as we move from the depolarization phase to the repolarization phase, those voltage-gated sodium ion channels, they close, right? And the voltage-gated potassium ion channels open up. So those voltage-gated sodium ion channels, they close and they enter into a resting phase. They cannot physically reopen for some very brief window of time. So in the absolute refractory period, we're talking about the phase of the action potential where either those voltage-gated sodium ion channels are already open and therefore cannot open more, or those voltage-gated sodium ion channels have just closed and they need to rest. The way the ion channel is built, it will not reopen for a brief window of time. So during the absolute refractory period, for these reasons, another action potential is absolutely not possible. Let's think about the relative refractory period. That's the sort of orangey tan region. So the relative refractory period begins during the sort of the end of the repolarization phase and continues through the hyperpolarization phase. So what's happening here? On the orange parts of this graph, remember voltage-gated potassium ion channels are open and potassium is flowing out of the cell. Potassium is flowing out, flowing out, flowing out, taking its positive charge with it, which means the inside of the cell is becoming increasingly negative, and that's why the line goes down on the graph. Well, the sodium ion channels at this point, they have rested. They're ready to open again. They're voltage-gated sodium ion channels, so they will open at a particular voltage. They will open when the membrane potential reaches threshold. When the membrane potential reaches -55, those voltage-gated sodium ion channels will open again and another action potential will be initiated. But this is very hard. It's a hard thing to do during the relative refractory period. It's very difficult to oppose those potassium ion channels that are leaving the cell, causing repolarization of the membrane. There's all this potassium leaving. How are we possibly going to get back up to threshold? We'd have to reverse that tide, so to speak. Okay, so during the absolute refractory period, another action potential is impossible, no matter how large the stimulus. But during the relative refractory period, an action potential could occur, but it's difficult because we need a very large stimulus to bring that membrane that is currently either repolarizing or hyperpolarizing. We need an extra big stimulus to bring it up to threshold at -55. All right, I believe I explained all this. It's written out in case you need it. Ooh, this is here to remind you of a great analogy. So at one semester in class, as I was explaining the absolute refractory period and the fact that the way the voltage gated sodium ion channels are built is such that once they close, they have to rest before they can reopen again. And this one woman raised her hand and she said, "Laura, my dad's a plumber. Is it like a toilet?" And I said, yes, it's exactly like a toilet. That's a great way to think about it. So we all know that after you flush the toilet, we can't flush the toilet again until it finishes flushing. And then the tank has to fill, right? So we've all tried to flush again mid-flush and it doesn't work. And then we've all waited a few seconds and tried to flush even before the tank refills with water. And we know that that doesn't work. So during the flush and while the tank is refilling is like the absolute refractory period. And voltage gated sodium ion channels are super cool the way they're built. This is a very simplified diagram. This doesn't show how cool they are. If you're curious, go and look them up. So for us, voltage gated sodium ion channels are either closed, opened, or resting. And then I believe I gave you all of this information about the relative refractory period a few slides ago. It's written out here if you need it. And our next topic is to talk about propagation of action potentials. So how do action potentials travel? They spread across a plasma membrane from one region to the next region. How does that happen? And so there are two different methods by which action potentials can spread or travel, or propagate is the proper word. How do action potentials propagate? And these two forms of propagation are called continuous and saltatory. Continuous conduction is the slower way for an action potential to propagate, and saltatory conduction is much faster. And we will cover the mechanism of each. So continuous conduction occurs in unmyelinated axons. So there are no myelin sheaths coating the exterior of the axon. And it's called continuous conduction because one segment of the plasma membrane depolarizes the next segment, depolarizes the next segment, I shouldn't say inch by inch. That's the wrong order of magnitude, but tiny little bit by tiny little bit. It's often likened to a domino effect, and an image is really a useful way for describing or understanding continuous conduction. So notice in the diagram up top, we are sort of mid-axon. What is happening as the action potential is going to pass like a wave through this segment of the axon? First, for any given location on that axon, depolarization will occur and then repolarization will follow. So down below, we see those two parallel lines and we see plus and minus signs to indicate what's happening on the inside of the cell relative to the outside of the cell. And in the blue portion of that axon, we see that depolarization is happening, where there's an influx of positively charged sodium ions, and that causes the plasma membrane in that region to depolarize. Well, those sodium ions sort of spread out once they're inside the cell. So this local influx of sodium ions causes the neighboring region of the plasma membrane to also depolarize, to reach threshold. And guess what happens when the neighboring plasma membrane reaches threshold? Those voltage-gated sodium ion channels open up, and so on and so on. So if we follow the progression of an action potential down that segment of the axon, up top we see what we were just looking at in the previous slide. We see depolarization in one region of the plasma membrane, and if we now move on to the middle panel there, that causes depolarization of the next region of the plasma membrane. And notice that depolarization is followed by repolarization. And if we go from the middle panel to the bottom panel, the same sequence of events is occurring. One segment of the plasma membrane depolarizes, and that is sufficient to cause depolarization in the next segment. So those voltage-gated sodium ion channels open up. All right, saltatory conduction is a little bit different. Saltatory conduction occurs in myelinated axons. So myelin is this cool stuff. It's often described as a fatty substance that wraps around the exterior of axons. And myelin is actually created by a cell that wraps itself like almost like a super thin pancake or a crepe around the axon. So looking on the right side of this diagram, we see that yellow axon and we see two myelin sheaths there, those sort of like sausage looking white things. And then we can see the nucleus of that cell called a Schwann cell. So in the peripheral nervous system, myelin is made up of a type of glial cell called Schwann cells. And these Schwann cells, during development, these Schwann cells grow in such a way that they wrap themselves around the axon. So what myelin really is like layers and layers and layers of plasma membrane. And plasma membrane is made out of phospholipids for the most part. Yeah, there's proteins embedded in it and some cholesterol, a few other things. But that's why myelin is often described as a fatty coating, because it's membrane, it's plasma membrane, layers and layers of it, lots and lots of phospholipids. Okay, in between neighboring myelin sheaths are these gaps called nodes of Ranvier, some French guy. And those nodes are really important for understanding what happens during saltatory conduction. So if we go to the left and we just look at that whole axon, we see myelin sheath, node of Ranvier, myelin sheath, node of Ranvier, myelin sheath, node of Ranvier. So that alternation is important as well. Let's take a closer look at what happens during saltatory conduction. Oh, let's take a closer look at a cross-section through an axon that is myelinated. So in the middle there, we see the axon, and we see probably those little smaller gray circles are probably mitochondria. And then on the outside of the cell, those darker sort of concentric circles, those are layers of myelin. And we can even see on the right side, we can even see the nucleus of the Schwann cell. So it's a super cool image. This is a scanning electron micrograph. All right, so saltatory conduction occurs in myelinated axons. And the reason why it can happen so much faster is because depolarization really only occurs at the nodes. The voltage-gated sodium ion channels and the voltage-gated potassium ion channels are super duper condensed at the nodes. So it's a very high intensity event there. Here we're looking at a myelinated axon where a section of the axon as well as the myelin sheaths have been sliced away longitudinally, so we can see what's going on underneath the myelin and at the nodes of Ranvier, so in between adjacent myelin sheaths. So myelin is sometimes described as an insulator because the regions of plasma membrane deep to the myelin do not contain very many voltage-gated ion channels. Instead, the voltage-gated ion channels, both sodium and potassium, are concentrated at the nodes. So all of the conduction, all of the flow of charged particles happens at the nodes of Ranvier. And they are very dramatic events because there are just so many ion channels there and they all open at once. So all the voltage-gated sodium ion channels open up and there's this massive influx of sodium ions. And once those sodium ions move into the cell by facilitated diffusion, they will diffuse in the interior of the cell. They'll move away from the node, from an area of high concentration to an area of low concentration, and they'll diffuse away from the node all the way through the axon to the next node, where those charged particles, those sodium ions,
are in sufficient quantity to cause a local depolarization at the next node. And all that node has to do is reach negative 55 millivolts. And guess what? All the voltage-gated sodium ion channels open there. And so saltatory conduction is a type of action potential propagation where the action potential jumps from node of Ranvier to node of Ranvier. And if you're a Spanish speaker, or maybe you've learned a little bit of Spanish, saltar, what does that mean? It means to jump. So it's saltatory conduction. And I don't know if you're familiar with the tale of the tortoise and the hare. But in this fable, this parable, this story, a rabbit and a turtle are in a race with each other. And the rabbit is very arrogant. He's very boastful. And the turtle is very humble, but slow and steady. And so the turtle, think about the ground that the turtle has to cover to get from the start line to the finish line, and think about the ground that a rabbit has to cover in order to get from the start line to the finish line. The turtle has to pretty much touch every piece of ground from the start line to the finish line, slow and steady. That's continuous conduction. But the rabbit can leap over large chunks of ground from between the start line and the finish line. That is saltatory conduction, and it occurs faster than continuous conduction. And saltatory conduction occurs in myelinated neurons. As you already know, neurons communicate with other neurons at synapses. They also communicate with their effectors at synapses, right? Like the neuromuscular junction is an example of a synapse, only it's between two different types of cells, a neuron and a muscle cell. Here we're looking at a synapse between two neurons. And there's the presynaptic cell and the postsynaptic cell. So we're going to revisit synapses briefly. And we have already studied the events that occur at a chemical synapse. At a chemical synapse, the message is transmitted from the presynaptic cell to the postsynaptic cell via a neurotransmitter, a chemical signaling molecule. There are also electrical synapses where there is no neurotransmitter. And let me use a picture to show you the differences between a chemical synapse and an electrical synapse. So on the left, we see a chemical synapse, and you are familiar with the structures here. Up top, we have the presynaptic cell. Down below, we have the postsynaptic cell. The presynaptic cell, in response to an action potential, will release neurotransmitter by exocytosis, and that neurotransmitter binds to neurotransmitter receptors in the postsynaptic cell. On the right, we see what's known as an electrical synapse, and there is no synaptic gap here. Rather, the plasma membranes of the two cells, the presynaptic neuron and the postsynaptic neuron, their plasma membranes are physically joined together by a type of cell junction called a gap junction. And these gap junctions are fluid-filled tunnels. So the cytosol of one cell is directly linked to the cytosol of another cell. So if you're a sodium ion in the cytosol of the presynaptic cell, you can easily diffuse through the gap junction into the cytosol of the postsynaptic cell. So when an action potential arrives at the axon terminal of the presynaptic cell and sodium ions are rushing in to the axon terminal, those sodium ions diffuse in all directions, but some of them will go through those gap junctions and enter the postsynaptic cell, causing local depolarization there. In the postsynaptic cell, all we need to do is hit negative 55 millivolts and an action potential will happen. Here we see a different representation of an electrical synapse. And notice here, we're not even talking about neurons. This happens to be an electrical synapse between two smooth muscle cells, which we haven't really talked about the physiology of smooth muscle. We've talked about the physiology of skeletal muscle, but it's a great image to demonstrate what an electrical synapse is. And so we can see how the two plasma membranes are joined together by those gap junctions, and those gap junctions are tunnels. They are connections between the cytosol of one cell and the cytosol of the next cell. And we see positively charged ions flowing from one cell to the next. And those positively charged ions will cause a change in membrane potential. All right, so this brings us to the end of the first segment of 5 segments on the nervous system. And today we talked about topics number 1 through topic number 6. And in the next segment, segment 2, we'll pick up at topic number 7. I will see you there.